Health authorities and genomic researchers are currently analyzing a significant development in the tracking of rodent-borne pathogens. Following a series of fatalities linked to a rare viral strain, the focus of the international public health community has shifted toward a cluster of cases involving the Andes virus, a specific type of orthohantavirus primarily found in South America. This investigation gained urgency following a multi-country outbreak associated with an international cruise vessel, the MV Hondius, which departed from Ushuaia, Argentina, on April 1, 2026. The voyage, which carried 147 passengers and crew from 23 different nations, was intended to traverse the South Atlantic with stops at remote locations, including Antarctica, South Georgia Island, and St. Helena. However, the mission transformed into a public health emergency as passengers began exhibiting symptoms of a severe respiratory nature. The index case, a passenger who developed initial symptoms of fever and gastrointestinal distress on April 6, died while on board on April 11. This initial fatality was followed by a sequence of similar clinical presentations among other travelers, prompting a coordinated response from the World Health Organization and the Africa Centers for Disease Control and Prevention. +5 Shuptrat-Crete, the biological mechanism driving these fatalities, is identified as Hantavirus Pulmonary Syndrome, or HPS. In clinical terms, the virus targets the vascular endothelial cells, which form the lining of the blood vessels. When the virus infects these cells, it triggers an intense and often dysregulated immune response. Rather than the virus destroying the tissue directly, the body's own inflammatory signals cause the capillaries in the lungs to become highly permeable. This leads to what clinicians describe as capillary leak, where fluid from the bloodstream floods the air sacs of the lungs. The progression is notably rapid. After an initial prodromal phase of fever, muscle aches, and headaches, patients can enter the cardiopulmonary phase within hours or days. This phase is characterized by the sudden onset of pulmonary edema, acute respiratory distress, and shock. Because the lungs essentially fill with fluid, the capacity for oxygen exchange is severely compromised, leading to the rapid respiratory failure described by researchers as the shutting down of the respiratory system. Unlike many other hantaviruses found in Europe and Asia, which primarily affect the kidneys and cause hemorrhagic fever with renal syndrome, the New World strains found in the Americas, such as the Andes virus, are specifically associated with this high fatality pulmonary outcome. Current data indicates that mortality rates for Andes virus infections typically range between 30 and 60%, making it one of the most lethal respiratory pathogens currently monitored by global surveillance networks. The investigation into the recent outbreak has moved into a high-intensity laboratory phase, as scientists attempt to decode the specific genomic profile of the virus responsible for the cruise ship fatalities. Genomic sequencing, conducted by the National Institute for Communicable Diseases in South Africa, in collaboration with the Geneva University Hospitals and the University of Zurich, has provided the first clear look at the pathogen's molecular structure. The hantavirus genome is composed of 3 segments of single-stranded, negative-sense RNA, labeled as the large, medium, and small segments. Researchers focusing on the large segment, which encodes the viral RNA-dependent RNA polymerase, have identified specific mutations that distinguish this 2026 strain. While the sequences show a high degree of identity, exceeding 98.7% to historical strains detected in Argentina in 1997 and 2018, the presence of specific synonymous mutations and variations in the L segment has drawn significant interest. These findings suggest that the outbreak most likely originated from a single zoonotic spillover event, where the virus jumped from a rodent reservoir to a human host, followed by a limited chain of transmission. +1A critical factor in this genome mystery is the confirmation that the pathogen is indeed the Andes virus. This distinction is vital for public health logistics because the Andes virus is the only hantavirus currently known to be capable of sustained, albeit limited, person-to-person transmission. Most hantaviruses require direct or indirect contact with infected rodents, typically through the inhalation of aerosolized droppings, urine, or saliva in enclosed spaces. However, The Andes virus can spread through close physical contact or exposure to the respiratory secretions of an infected individual. This unique characteristic explains why the outbreak on the MV Hondius was treated with extreme caution by international maritime and health agencies. In early May 2026, as the ship reached the Canary Islands, the World Health Organization confirmed 6 laboratory-verified cases and 2 suspected cases. The logistical challenge was immense. The body of the index case had been removed at St. Helena, while a second patient collapsed at an airport in Johannesburg and died shortly after. A third patient remained in critical condition in a South African intensive care unit. Laboratories are now contrasting these confirmed cases against the reported symptoms of other passengers to determine the full extent of the silent or mild infections that may have occurred during the voyage. As early sequencing suggests, the virus may have been circulating on the vessel for several weeks before the full scale of the danger was realized. In response to the identified risk, a coordinated international surveillance and prevention effort has been activated. On May 11, 2026, the controlled evacuation of all passengers and crew from the MV Hondius was completed in Granadilla, Tenerife. The operation utilized bio-isolation pods and high-level personal protective equipment to prevent any further environmental or person-to-person spread during the transit of passengers back to their respective home countries. The Pan American Health Organization has issued updated guidance for the southern cone of South America, where the Andes virus is endemic, noting that while the cruise ship cluster is an isolated event, environmental changes are shifting the range of the virus's natural hosts. The primary reservoir for the Andes virus is the long-tailed pygmy rice rat, a small rodent whose population dynamics are heavily influenced by climate and food availability. Public health officials emphasize that prevention remains the most effective tool, as there are currently no widely approved vaccines or specific antiviral treatments for hantavirus infection. Current medical management is restricted to supportive care, often involving mechanical ventilation and intensive fluid management to navigate the critical pulmonary phase. +4 surveillance programs are now being expanded to include more rigorous testing of wildlife populations and increased monitoring of rural and peri-urban areas where human-rodent contact is most frequent. In Europe, the National Reference Center for Hantaviruses at the Pasteur Institute has noted a steady number of cases consistent with seasonal averages. But the 2026 South Atlantic cluster has prompted the activation of a Level 1 outbreak response unit by the ANRS Emerging Infectious Diseases Agency. This unit is tasked with defining research priorities, including the development of subunit vaccines that target the viral surface glycoproteins, known as GN and GC. These proteins form the spike complex that allows the virus to enter human cells. By creating vaccines that mimic these structures, researchers hope to induce an immune response that can block the infection before it reaches the lungs. While the immediate threat from the cruise ship cluster appears to be contained through quarantine and isolation, the limits of current scientific knowledge regarding the evolutionary trajectory of the Andes virus remain a point of concern. Health agencies continue to urge travelers and residents in endemic areas to avoid activities that stir up dust in areas where rodents may nest, and to seek immediate medical attention if fever is followed by any difficulty breathing. Subscribe for more in-depth reports on emerging health security and global viral trends. The current laboratory investigation into the reported genomic mystery centers on the molecular structure of the orthohantavirus genome, which consists of 3 segments of single-stranded RNA. These segments, designated as small, medium, and large, encode the nucleocapsid protein, the envelope glycoproteins, and the viral RNA polymerase, respectively. According to protocol established by the Centers for Disease Control and Prevention and the World Health Organization, any suspected variation in these segments requires immediate confirmation through metagenomic next-generation sequencing. Scientists are specifically scrutinizing the medium segment, which dictates the structure of the G and GC glycoproteins. These proteins are responsible for the virus's ability to bind to cellular receptors, specifically the beta-3 integrins found on human endothelial cells. A mutation in this region could theoretically alter the virus's affinity for these receptors, potentially accelerating the speed at which the virus enters the bloodstream and targets the pulmonary system. In high-containment biosafety level 4 laboratories, researchers are using reverse genetics to isolate these specific mutations and determine if they correlate with the accelerated clinical decline reported in recent cases. The diagnostic process for identifying this specific hantavirus strain involves a multi-tiered testing approach. Initial screening typically utilizes enzyme-linked immunosorbent assays, or ELISA, to detect the presence of IgM and IgG antibodies, which indicate a recent or past infection. However, because these antibodies may not appear until several days after the onset of symptoms, molecular testing via reverse transcription polymerase chain reaction, known as RT-PCR, is critical for early detection. RT-PCR allows clinicians to identify the presence of viral RNA in the blood or tissue samples during the early prodromal phase. National reference laboratories are currently comparing sequences from the most recent fatalities against historical data stored in the GenBank database to identify any significant drift or shift in the viral genome. While the source headline suggests an instant shutdown of the respiratory system, virologists clarify that the biological mechanism involves a complex cascade of events. The virus does not directly destroy the lung tissue. Instead, it triggers an intense inflammatory response known as a cytokine storm. This leads to the sudden increase in vascular permeability, where fluid from the capillaries leaks into the alveolar spaces, resulting in acute pulmonary edema. This clinical progression, while rapid, is the focus of intense scrutiny to determine if a specific mutation has shortened the time frame between the initial fever and the onset of respiratory failure. International health agencies and hospital networks are currently on high alert, standardizing the criteria for identifying suspected cases. The World Health Organization's Global Outbreak Alert and Response Network is coordinating with regional clinics to ensure that diagnostic samples are handled under strict Category A infectious substance regulations. This requires specialized packaging and logistics to prevent environmental exposure during transport to specialized BSL-4 facilities, such as those operated by the National Institute of Allergy and Infectious Diseases. Hospitals are being advised to monitor for the triad of Hantavirus Pulmonary Syndrome symptoms: fever, a drop in platelet count, known as thrombocytopenia, and the presence of immunoblasts or atypical lymphocytes in the blood. While laboratory findings are still being verified, officials are contrasting the sensational claims of an instant virus with the reality of known viral mechanisms, emphasizing that even a mutated strain must follow the biological laws of viral replication and host response. The focus remains on whether the observed mutation facilitates a more efficient bypass of the human innate immune system, specifically the interferon response, which normally serves as the body's first line of defense against viral invasion. In the wake of recent genomic findings, global health agencies are emphasizing a dual strategy of aggressive environmental surveillance and refined public prevention protocols. The primary challenge in controlling hantavirus, particularly the Andes and Sin Nombre strains, lies in its ecological persistence within wild rodent reservoirs. Surveillance teams from the Pan American Health Organization and the European Center for Disease Prevention and Control are currently expanding One Health monitoring programs. These programs involve trapping and testing local rodent populations, specifically the long-tailed pygmy rice rat in South America and the deer mouse in North America, to measure viral seroprevalence. By tracking the percentage of infected animals in a specific geography, officials can issue early warnings to rural and suburban communities before a spillover event occurs. This data-driven approach is critical because hantavirus outbreaks are often linked to environmental fluctuations, such as increased rainfall or masting events, where an abundance of seeds leads to a population explosion among host rodents. When these populations surge, the likelihood of human-rodent interaction increases, raising the statistical probability of aerosolized transmission. Public health guidance remains focused on the mechanics of transmission to prevent the inhalation of viral particles. Experts stress that hantaviruses are fragile outside the host and are easily deactivated by common disinfectants, yet they remain highly dangerous when disturbed in dry environments. The standard protocol for cleaning potentially infested areas, such as cabins, sheds, or storage units, strictly forbids the use of vacuums or brooms, which can loft viral particles into the air. Instead, the wet cleaning method is mandated, saturating the area with a 10% bleach solution or a high-concentration disinfectant for at least 5 minutes before wiping. This process ensures that any viral matter present in rodent excreta is chemically neutralized and physically weighed down, preventing the formation of the infectious aerosols that target the human respiratory system. For individuals in high-risk professions, such as park rangers, construction workers in rural areas, or maritime staff handling cargo, the use of N95 or higher-rated respirators is increasingly recommended as a standard safety measure when working in enclosed, poorly ventilated spaces where rodent activity is suspected. The limits of current medical knowledge present a significant hurdle for clinical response, as there is currently no specific antiviral therapy or vaccine approved for Hantavirus Pulmonary Syndrome. Clinical management is essentially a race against the body's inflammatory response. In severe cases, the only viable intervention is extracorporeal membrane oxygenation, or ECMO, a highly resource-intensive process where a machine takes over the function of the heart and lungs to maintain oxygenation while the patient's own organs recover from the capillary leak phase. However, ECMO is not a cure and is only available in advanced tertiary care hospitals, creating a significant disparity in survival rates between urban centers and the remote areas where the virus is most frequently encountered. Researchers are currently investigating the efficacy of neutralizing antibodies and broad-spectrum antivirals, but these remain in the experimental or trial stages. This gap in treatment underscores the necessity of early detection. The transition from the prodromal phase of fever and aches to the critical respiratory phase can occur in as little as 24 to 48 hours, leaving a narrow window for medical intervention. As the scientific community continues to investigate the reported genomic mutations, the ethical imperative for clear, non-sensational reporting becomes paramount. While the term genome mystery captures public attention, virologists note that all RNA viruses undergo constant evolutionary drift. The task for researchers is to determine if these specific genetic changes actually translate to increased virulence, or if the perceived instant shutdown of the lungs is a reflection of the virus's known biological baseline in vulnerable individuals. Public health officials urge a balanced perspective, acknowledging the high fatality rate of hantavirus, while recognizing that it remains a rare disease that requires specific environmental conditions to spread. By maintaining rigorous surveillance of rodent hosts and adhering to established sanitation protocols, the risk to the general public remains statistically low. The ongoing work in genomic sequencing is not a cause for panic, but a tool for precision medicine and better diagnostic preparedness in a world where the boundaries between human habitats and wild reservoirs are increasingly blurred. For continued coverage of emerging viral threats and public health infrastructure, subscribe to this channel.